CSIRNA Life Sciences, DNA replication prokaryotes and eukaryotes. DNA replication is the process by which the entire DNA content of cells duplicated to ensure transmission of genetic information to other cells. It is a semi-conservative process. It is experimentally proven by Wilson et al., where each DNA molecule retains one parental strand and synthesizes one new strand. This mechanism is considered proximate to exist structural and regulatory differences between prokaryotes like E. coli and eukaryotes like human cells. Replication always occurs in the 5' to 3' direction, with synthesis catalyzed by DNA polymerases and deoxyribonucleotide triphosphates using an existing template strand. In prokaryotes, replication begins at a single origin of replication call which is 245 BP long ET region. Initiated protein DNA binds to the DNA boxes to begin melting with double helix. The helicase DNA B, loaded with the help of DNA C, unwinds the DNA of the replication fork. Single-stranded binding proteins SSB stabilize the unwound strands. Kinase DNA G synthesizes short RNA primers necessary for the initiation of DNA synthesis, since DNA polymerases cannot initiate synthesis de novo. DNA polymerase frees the principal replicative polymerase that extends the primer. It is a highly processed enzyme with sliding clamp, beta clamp that ensures rapid synthesis. The leading strand is synthesized continuously, while the lagging strand is synthesized continuously in no fragments, which are later joined by DNA oligosaccharides. DNA polymerase removes RNA primers via its 5' to 3' deoxyribonucleic activity and replaces them with DNA. DNA oligosaccharides seals the mix in a sugar phosphate backbone. Termination of replication in E. coli is facilitated by two-steric complexes that blocks the replication fork. In eukaryotes, the replication machinery is more complex due to linear and large-sized chromosomes. Multiple origins of replication exist along each chromosome to ensure timely duplication. Each origin is marked by the origin recognition complex fork, which binds certain G1 phases and recruits other proteins including CDC6, CDT1, and the chromosome maintenance MCM helicase complex to form the pre-replicative complex RC. Activation of this complex in S phase involves CDKs and DKs, which lead to recruitment of polymerases and addition of replication. Eukaryote polymerases are specialized. DNA polymerase alpha has kinase activity and synthesizes the RNA DNA hybrid primer. Polymerase delta extends the lagging strand, and polymerase epsilon synthesizes the leading strand. The sliding clamp in eukaryotes is pCNA, proliferating cell nuclear antigen. RNA primers are removed by RNase H and FEN1, flap and nuclease 1. DNA oligosaccharides seals the fragments. Replication ends with the problem of replication of chromosomes and telomeres. The enzyme telomerase, a ribonucleoprotide with reverse transcriptase activity, extends the telomere repeats to prevent loss of genetic information from the ends. Replication fidelity is maintained by the 3' to 5' deoxyribonucleic activity of DNA polymerases, which excises matched nucleotides. Additionally, post-replication mismatch repair mechanisms scan synthesize DNA. In prokaryotes, mismatch repair uses a methylation pattern. Parental strand is methylated to distinguish the template from the new strand. In eukaryotes, the mechanism is more complex and uses an lagging strand and replication-associated proteins for strand discrimination. The regulation of replication differs significantly. In prokaryotes, replication is tightly coupled with the cell cycle, and DNA ATP levels control the timing of initiation. In contrast, eukaryotic replication is strictly coordinated with the cell cycle via checkpoints and cycling CDKs. Each origin fires only once for cell cycle due to licensing controls. Overreplication is prevented by degradation and inhibition of licensing proteins after initiation. Antibiotics and chemotherapeutic agents often target DNA replication. For example, ciprofloxacin inhibits prokaryotic DNA gyrase, a topoisomerase, while epithelial inhibits eukaryotic DNA polymerase alpha. Replication stress supports cell and leads to genome instability, which is a hallmark of cancer. Understanding replication mechanisms is crucial for developing targeted therapies and understanding mutagenesis. Previous year question from CSIRNA Life Sciences. 2017 June, Part C. Which one of the following statements is correct about DNA replication in E. coli? A DNA G synthesizes RNA primer in the 5' to 3' point arrow 3' direction. Answer. A DNA G synthesizes RNA primer in the 5' to 3' point arrow 3' direction. 2019 June, Part B. Which eukaryotic DNA polymerase is responsible for synthesis of the lagging strand? Answer. DNA polymerase delta. 2022 June, Part C. During lagging strand synthesis in eukaryotes, which enzymes are responsible for primer removal? Answer. RNase H and FEN1. Let me know the next topic you want to cover, or I can proceed in slow order. You said, inheritance biology 8, Mendelian principles, ChatGPT said. Absolutely. Here's an elaborate, high-quality lecture on Mendelian principles tailored for CSIRNA Life Sciences based on frequently asked questions and important concepts. This is written in paragraph form, maximum word limit, with no tables, and includes 2 to 3 previous year CSIR questions with answers and years mentioned at the end. This lecture is crafted to be your only coaching material, so it is complete from base to advanced level. CSIRNA Life Sciences, inheritance biology, Mendelian principles. Mendelian principles, the foundation of classical genetics, were established by Gregor Mendel through his experiments with Vincent Young, Gordon P. in the mid-19th century. His observations led to the formulation of 3 major laws: the law of segregation, the law of independent assortment, and the law of dominance. These laws laid the groundwork for understanding how genetic traits are inherited from one generation to the next and remain central to modern molecular genetics. The law of segregation, also called Mendel's first law, states that alleles of genes segregate from each other during gametes formation, ensuring that each game carries only one allele for each gene. This is supported psychologically by the separation of homologous chromosomes during anaphase and meiosis. Mendel deduced this principle through a monohybrid process, in which he crossed breeding plants differing in one trait, such as flower color e.g., purple times white. The F1 generation exhibited only the dominant trait purple, but the recessive trait white reappeared in F3, one ratio in the F2 generation, indicating that the alleles separated and recombined independently. The law of independent assortment, Mendel's second law, asserts that alleles of different genes assort independently when another during game formation, provided the genes located on different chromosomes or are far apart on the same chromosome. This was established through Mendel's hybrid process, where simultaneously examined the inheritance of 2 traits, such as speed, shape, and color. The F2 generation showed a 9:3:3:1 phenotypic ratio, supporting the idea that each gene pair separates independently. This law, however, has exceptions in cases where genes are linked, i.e., located close together on the same chromosome, which violates the principle of independent assortment due to physical linkage. The law of dominance, although not formally stated by Mendel's law, explains that in heterozygote, one allele can match the expression of the other. The expressed allele is termed dominant, and the masked allele is termed recessive. This was observed in Mendel's experiments when the F1 offspring always exhibited a dominant phenotype. The law of segregation, also called Mendel's first law, states that alleles of genes segregate from each other during game formation, ensuring that each game carries only one allele for each gene. This is supported psychologically by the separation of homologous chromosomes during anaphase and meiosis. Mendel deduced this principle through a monohybrid process, in which he crossed breeding plants differing in one trait, such as flower color e.g., purple times white. The F1 generation exhibited only the dominant trait purple, but the recessive trait white reappeared in F3, one ratio in the F2 generation, indicating that the alleles separated and recombined independently. The law of independent assortment, Mendel's second law, asserts that alleles of different genes assort independently when another during game formation, provided the genes are located on different chromosomes or are far apart on the same chromosome. This was established through Mendel's hybrid process, where simultaneously examined the inheritance of 2 traits, such as speed, shape, and color. The F2 generation showed a 9:3:3:1 phenotypic ratio, supporting the idea that each gene pair separates independently. This law, however, has exceptions in cases where genes are linked, i.e., located close together on the same chromosome, which violates the principle of independent assortment due to physical linkage. The law of dominance, although not formally stated by Mendel's law, explains that in heterozygote, one allele can match the expression of the other. The expressed allele is termed dominant, and the masked allele is termed recessive. This was observed in Mendel's experiments when the F1 offspring always exhibited a dominant phenotype, regardless of the presence of a recessive allele. It is important to note that dominance is a relative concept and not universal. Some traits show incomplete dominance or dominance, such as the pink flower color in snapdragons, incomplete dominance, or E. blood group in humans co-dominance. The application of Mendelian principles extended through Planck squares and probability calculations to predict the outcomes of genetic process. The genotype and phenotypic ratios predicted by these laws can be altered by gene interactions such as epistasis, incomplete dominance, co-dominance, and multiple alleles. Moreover, deviations from Mendelian ratios may occur due to linkage, gene lethality, penetrance, expressivity, and cytoplasmic inheritance. Molecular validation of Mendel's law came with the discovery of genes and chromosomes. The chromosomal theory of inheritance, proposed by Sandra Berry, integrated Mendel's principles with psychological observations, confirmed that genes reside on chromosomes and their behavior during meiosis explains the patterns of inheritance. This was supported by studies in Drosophila melanogaster by Thomas Morgan, who demonstrated excellent inheritance and recombination, providing evidence for linkage and crossing over. Mendelian principles have been further expanded in the context of human genetics, especially the inheritance patterns of monogenic disorders such as cystic fibrosis, autosomal recessive Huntington's disease, autosomal dominant, and hemophilia A X linked recessive. In pedigrees, Mendelian inheritance allows prediction of carrier status, risk of disease transmission, and understanding hereditary patterns. Modern molecular tools, such as PCR-based genotyping, DNA sequencing, and CRISPR-Cas9, allow precise tracking and manipulation of Mendelian traits. Despite the complexity introduced by polygenic traits, epigenetic factors, and environmental influences, the Mendelian framework remains essential for exact genetic architecture and inheritance patterns. For CSIRNet JRF Life Sciences, a thorough understanding of mental principles and their acceptance is vital, as these principles not only form core components of facilities, but also appear frequently in conceptual, application-based questions. Previous year CSIRNet questions. 2022 June, Part B. In Mendelian inheritance, what is the expected phenotypic ratio of offspring from a hybrid cross AAVB times AAVB, assuming independent assortment? Answer, 9, 3, 3, 1. 2021 June, Part C. A monohybrid cross between two heterozygous individuals yields a phenotypic ratio of 3, 1 in F2 generation. What is the genotypic ratio? Answer, 1, 2, 1, A, A, ah, ah. 2017 December, Part C. Which of the following does not conform to Mendel's law of independent assortment? Answer, linked genes on the same chromosome do not assort independently. Let me know if you'd like to continue to the next subtopic under inheritance biology, e.g., chromosomal theory of inheritance or gene interactions, or suggest your next topic of choice. You said, dominance, ChatGPT said. Here's your maximum detail. High quality CSIRNet JRF Life Sciences lecture on dominance, written in dense paragraph form, using the maximum word limit, no tables, and practice exercises to the CSIRNet JRF exam level. This includes 2 to 3 previous year questions with answers and yours as well. This is your only study material in coaching, so learning essentials covered with no free space wasted. CSIRNet JRF Life Sciences, inheritance biology, dominance. Dominance is a fundamental genetic principle that describes the relationship between alleles of gene expression and the phenotype of heterozygotes. When an organism carries two different alleles of a given locus, the allele that is phenotypically expressed is called the dominant allele, while the one that is masked is called the recessive allele. This classical concept, first observed by Mendel in his experiments with pea plants, is a simplified representation of a more complex spectrum of allelic interactions observed in modern genetics. Dominance is not an inherent property of an allele, the relationship between the alleles of the same locus can be complete, incomplete, or codominant, depending on how the heterozygote phenotype compares to those of the homozygotes. Complete dominance occurs when the dominant allele completely masks the effect of the recessive allele in heterozygotes. For example, in Mendel's pea plant experiments, the allele for purple flower color was completely dominant over the allele for white flower color, leaving all heterozygotes have one individual to display the purple phenotype. In this case, the phenotypic ratio of a monohybrid crossing F2 generation is 3, 1, while the genotypic ratio remains 1, 2, 1. Although this appears simple, the molecular basis often involves a dominant allele encoding functional protein, such as the pigment enzyme, whereas the recessive allele is usually loss of function mutation. In contrast, incomplete dominance refers to a situation where the heterozygote phenotype is intermediate between two homozygotes, indicating that either allele is completely dominant over the other. A classic example is the flower color in snapdragons and dry matches, where red flower homozygote crossed with white flower homozygote produces pink flowers in F1 generation. The F2 generation then displays the phenotypic genotypic ratio 1, 2, 1 red, pink, white revealing the heterozygote express the phenotype not seen in either parent. Incomplete dominance usually results when the amount of gene product is proportional to the number of functional alleles. Codominance occurs when both alleles in a heterozygote are fully simultaneously expressed without one masking the other. A textbook example is the human blood group system, where individuals with genotype IID express both N, B antigen versus red blood cells. Another example includes coat color in cattle, where red and white homozygotes produce one coded offspring when crossed, expressing both red and white hair. Codominance is often confused with incomplete dominance, but is distinct in that both phenotypes are fully present, not blended. The molecular mechanisms underlying dominance vary. In many cases, dominance arises from haploinsufficiency, where one copy of a functional allele produces enough protein to generate a normal phenotype, making the wild type allele over a non-functional mutant. Conversely, haploinsufficiency occurs when one copy is not enough for normal function, often resulting in a disease allele. Dominant negative mutations produce mutant proteins that interfere with the function of the wild type protein, as observed in disorders like Marfan syndrome. In gain of function mutations, the mutant allele acquires a new activity or is expressed inappropriately, leading to dominance, such as in Huntington's disease. Dominance is a fundamental genetic principle that describes the relationship between alleles of gene expression and the phenotype of heterozygotes. When an organism carries two different alleles of a given locus, the allele that is phenotypically expressed is called the dominant allele. When an organism carries two different alleles of a given locus, the allele that is phenotypically expressed is called the dominant allele, while the one that is masked is called the recessive allele. This classical concept, first observed by Mendel in his experiments with pea plants, is a simplified representation of a more complex spectrum of allelic interactions observed in modern genetics. Dominance is not an inherent property of an allele, the relationship between alleles of the same locus can be complete, incomplete, or codominant, depending on how the heterozygote phenotype compares to those of the homozygotes. Complete dominance occurs when the dominant allele completely masks the effect of the recessive allele in heterozygotes. For example, in Mendel's pea plant experiments, the allele for purple flower color was completely dominant over the allele for white flower color, leaving all heterozygotes have one individual to display the purple phenotype. In this case, the phenotypic ratio of a monohybrid crossing F2 generation is 3, 1, while the genotypic ratio remains 1, 2, 1. Although this appears simple, the molecular basis often involves a dominant allele encoding functional protein, such as the pigment enzyme, whereas the recessive allele is usually loss of function mutation. In contrast, incomplete dominance refers to a situation where the heterozygote phenotype is intermediate between the two homozygotes, indicating that neither allele is completely dominant over the other. A classic example is the flower color in snapdragons and dry matches, where red flower homozygote crossed with white flower homozygote produces pink flowers in F1 generation. The F2 generation then displays the phenotypic and genotypic ratio 1, 2, 1 red, pink, white revealing the heterozygotes express the phenotype not seen in either parent. Incomplete dominance usually results when the amount of gene product is proportional to the number of functional alleles. Codominance occurs when both alleles in a heterozygote are fully simultaneously expressed without one masking the other. A textbook example is the human blood group system, where individuals with genotype IID express both N, B antigens on the surface of red blood cells. Another example includes coat color in cattle, where red and white homozygotes produce one coded offspring when crossed, expressing both red and white hair. Codominance is often confused with incomplete dominance, but is distinct in that both phenotypes are fully present, not blended. The molecular mechanisms underlying dominance vary. In many cases, dominance arises from haploinsufficiency, where one copy of a functional allele produces enough protein to generate a normal phenotype, making the wild type allele dominant over a non-functional mutant. Conversely, haploinsufficiency occurs when one copy is not enough for normal function, often resulting in a dominant disease allele. Dominant negative mutations produce mutant proteins that interfere with the function of the wild type protein, as observed in disorders like Marfan syndrome. In gain of function mutations, the mutant allele acquires a new activity or is expressed inappropriately, leading to dominance, such as in Huntington's disease. Dominance can also be context dependent, where its expression varies due to genetic background, environment, or epistatic interactions. For example, a dominant allele in one genetic background may not exhibit dominance in another due to modifier genes. Environmental factors such as temperature and nutrition can modulate the dominance relationship, leading to phenotypic plasticity. Additionally, gene dosage and printing and epigenetic modifications can influence dominance patterns, making this a dynamic rather than static concept. In population genetics, dominance affects the rate at which alleles change in frequency under natural selection. Recessive deleterious alleles can persist in populations with low frequency because they are hidden in heterozygotes and thus not subject to selection. On the other hand, dominant beneficial mutations can spread rapidly, while dominant deleterious mutations are more efficiently purged. The dominance coefficient H is used to model these dynamics, where H equals 0 represents complete recessiveness, H equals 1 complete dominance, and H equals 0.5 partial or codominance. Understanding dominance is essential in areas like disease genetics, breeding, molecular biology, and biotechnology. Understanding dominance is essential in areas like disease genetics, breeding, molecular biology, and biotechnology. In human disease, dominant inheritance is typical in autosomal dominant disorders, where only one mutant allele is sufficient to express the trait. In plant and animal breeding, knowledge of dominance enables hybrid gear exploitation through heterosis. In molecular biology, dominant selection markers are used in gene editing and transgenic experiments to identify transformed cells. For the CSIRNet JRF Life Sciences exam, students must understand not only the basic definitions of dominance types but also their molecular basis, exceptions, and applications. Questions frequently appear testing whether students can distinguish between complete, incomplete, and codominance, interpret phenotypic and genotypic ratios, and analyze molecular data to explain allelic interactions. One must also be prepared to assess dominance in the context of population genetics and gene regulation. Previous year CSIRNet questions. 2018 December, Part B. Which of the following best describes predominance? Answer. Both alleles are expressed equally in the heterozygote. 2016 June. Part C. In a cross between a red flower and a white flower plant, all F1 individuals have pink flowers. What type of inheritance does this represent? Answer. Incomplete dominance. 2022 June. Part C. A heterozygous individual carrying one functional and one allele for a gene shows a normal phenotype. What is the molecular explanation? Answer. The gene product from one functional allele is sufficient. Haplosufficiency leading to complete dominance. Let me know if you'd like the next topic under inheritance biology, such as epistasis, gene interactions, or chromosomal basis of inheritance. I'm ready to build a full series for you, lecture by lecture, at the highest quality. You said, segregation, ChatGPT said. Here's your maximum detailed CSIR NetJRF Life Sciences lecture on segregation, part of inheritance biology. It is written in dense paragraph format, no tables, with maximum word limit, based on previously asked questions and important CSIR net concepts. This note is your only study material, so everything necessary to crack the exam is included, with no wasted space and deep conceptual coverage. CSIR NetJRF Life Sciences, inheritance biology, segregation. Segregation is one of the foundational principles of classical genetics and forms the core of Mendel's first law, the law of segregation. It states that during the formation of gametes, the two alleles for a gene located on homologous chromosomes separate from each other so that each gamete receives only one allele of each gene. This ensures that offspring inherit one allele from each parent for any given gene. The law was formulated through Mendel's monohybrid process in Piston sativa, wherein demonstrated that traits did not blend but rather remained discrete and segregated predictably through generations. This principle is groundbreaking because it introduced the idea of particular inheritance, in contrast to the previously accepted linear inheritance theory. Segregation is one of the foundational principles of classical genetics and forms the core of Mendel's first law, the law of segregation. It states that during the formation of gametes, the two alleles for a gene located on homologous chromosomes separate from each other so that each gamete receives only one allele of each gene. Segregation is one of the foundational principles of classical genetics and forms the core of Mendel's first law, the law of segregation. It states that during the formation of gametes, the two alleles for a gene located on homologous chromosomes separate from each other so that each gamete receives only one allele of each gene. This ensures that offspring inherit one allele from each parent for any given gene. The law was formulated through Mendel's monohybrid process in Piston sativa, wherein demonstrated that traits did not blend but rather remained discrete and segregated predictably through generations. This principle is groundbreaking because it introduced the idea of particular inheritance, in contrast to the previously accepted linear inheritance theory. At the molecular level, segregation is a direct consequence of meiosis, particularly during anaphase I, when homologous chromosomes each carrying one allele of a gene are pulled to opposite poles. This ensures that each gamete carries only one allele for each locus. If an individual is heterozygous for a gene, e.g., half of the gametes will carry the allele and the other half will carry the allele. The fusion of gametes through fertilization and source cycloidy, allowing for recombination of alleles in offspring. The random nature of segregation explains the predictable genotypic and phenotypic ratios in Mendelian processes. For a monohybrid process involving complete dominance, the X2 generation typically shows a 1, 2, 1 genotypic ratio AA, and 3, 1 phenotypic ratio, as C is dominant over A. Importantly, segregation is independent of the phenotype, meaning that even if one allele is phenotypically dominant, both alleles are equally likely to be passed on to the next generation. This insight is crucial in understanding carrier states in recessive disorders, where heterozygotes may transmit a recessive allele without expressing the phenotype. Non-disjunction during meiosis, which results in the failure of chromosome pairs to segregate properly, highlights the importance of accurate segregation. This error leads to aneuploidies, such as trisomy 21 Down syndrome or monosomy X Turner syndrome. These exceptions provide real-world evidence of how crucial precise segregation is to normal development. In CSIR net level questions, understanding segregation in the context of meiotic behavior is vital, especially in identifying whether observed offspring ratios deviate due to segregation distortion, chromosomal anomalies, or gene linkage. Segregation also applies to segregation distortion or meiotic drive, a phenomenon where certain alleles are transmitted to offspring more frequently than expected by Mendelian laws. These disorders may arise due to selfish genetic elements that manipulate the meiotic machinery to favor their own transmission. Such deviations from equal segregation are often seen in Drosophila, mice, and fungal species, and these are of intense research interest because they can impact population allele frequencies and evolution. Students should understand that segregation distortion is a violation of Mendel's law, often caused by selective gamete death, centromere competition, or sperm competition. Another layer is added by sex chromosomes, especially in heterogonetic sexes X, Y, or ZW, where the segregation of sex chromosomes determines the sex of the offspring. In mammals, for example, the segregation of X and Y chromosomes during In mammals, for example, the segregation of X and Y In mammals, for example, the segregation of X and Y chromosomes In mammals, for example, the segregation In mammals, for example, the segregation of X and Y chromosomes during spermatogenesis leads to 50% sperm carrying X and 50% carrying Y, giving an approximately equal male-female sex ratio. However, segregation bias in sex chromosomes, e.g., due to mutations in meiotic genes or meiotic drive elements, can skew sex ratios, which is a common topic in CSIR net and model organism questions. From an experimental point of view, segregation can be demonstrated using test crosses crossing a heterozygote with a homozygous recessive. A1. A1, 1 phenotypic ratio in progeny confirms. A1, 1 phenotypic ratio in progeny confirms segregation of alleles. Such experimental setups are classic in plant genetics and are frequently cited in net-level problems. Molecular tools like RFLP, SNP markers, and PCR-based genotyping now allow direct observation of allelic segregation at the DNA level, making classical segregation analysis relevant in modern molecular biology. Understanding segregation is not limited to single locus analysis. It is essential when solving genetic mapping problems, test cross analysis, epistasis, and linkage problems, all common in CSIR net JRF exams. It is also foundational to understanding Hardy-Weinberg equilibrium, population genetics, and disease prediction models. Thus, the law of segregation, while basic, is deeply integrated into advanced topics and must be mastered thoroughly. Previous year CSIR net questions, 2016 December, Part B. Mendel's law of segregation explains which of the following. Answer. During gamete formation, alleles of a gene segregate such that each gamete receives only one allele. 2018 June, Part C. In a cross between two heterozygotes, A times A, what is the expected genotypic ratio? Answer. 1, 2, 1 AA, A. 2021 February, Part C. Which meiotic stage ensures the segregation of alleles for a gene located on homologous chromosomes? Answer. Anaphase I. Would you like the next topic to be independent assortment, gene interactions, or linkage and recombination? I'm ready to build the entire inheritance biology unit at the same JRF cracking level. You said. Independent assortment. ChatGPT said. Here is your maximum detailed CSIR net JRF Life Sciences lecture on independent assortment, under inheritance biology, written in dense paragraph format, with no tables, maximum word limit, and zero wasted space. The content is tailored specifically to crack the CSIR net exam, from basic to advanced level, deeply focused on areas repeatedly asked in the exam. Includes 2 to 3 previous year questions with year and answers at the end. CSIR net JRF Life Sciences, inheritance biology, independent assortment. Independent assortment is a cornerstone of Mendel's second law, which postulates that genes located on different chromosomes assort independently of one another during the formation of gametes. This principle was first demonstrated by Gregor Mendel through dihybrid processes involving two traits, such as seed shape and seed color in Pisum sativum. From such experiments, he observed a 9:3:3:1 phenotypic ratio in the F2 generation when two heterozygotes for both traits were crossed. This ratio could only be explained if each pair of alleles separated independently of the other during gamete formation. Thus, Mendel deduced that the inheritance of one trait did not influence the inheritance of another, provided that the genes were not linked. At the chromosomal level, independent assortment is explained by the random orientation of homologous chromosome pairs on the metaphase I plate of meiosis. During metaphase I, each homologous chromosome pair aligns independently of all other pairs, allowing for a variety of possible combinations in the resulting gametes. This stochastic alignment results in genetic variation among offspring and is a primary contributor to the diversity produced by sexual reproduction. In a diploid organism with n pairs of chromosomes, there are two paired and possible combinations of chromosomes due to independent assortment alone. For humans, n equals 23. This accounts for more than 8 million combinations without even considering recombination. This principle, however, is strictly applicable only to genes located on different non-homologous chromosomes or genes that are sufficiently far apart on the same chromosome so that crossing over between them during prophase I meiosis allows them to assort independently. When genes are located close together on the same chromosome, they tend to be inherited together, a phenomenon known as linkage, which violates the principle of independent assortment. The deviation from the expected 9:3:3:1 ratio in such cases led to the discovery of genetic linkage and recombination mapping. The frequency of recombination between linked genes is used to estimate their distance apart on a chromosome, measured in centimorgans cm. Thus, understanding independent assortment is foundational for interpreting genetic linkage maps, which are frequently tested in CSIRNet life science questions. Independent assortment also provides a framework for solving test cross problems. For example, crossing a dihybrid AABB with a double recessive homozygote AABB produces a 1:1:1 ratio of phenotypes if the genes are independently assorting. Any significant deviation from this ratio can be attributed to linkage or segregation distortion. Such test crosses are routinely used in plant and animal breeding experiments to determine genotype and linkage relationships. Moreover, independent assortment is conceptually and mathematically linked to probability rules in genetics, such as the product rule multiplying probabilities of independent events and the sum rule adding probabilities of mutually exclusive events. These are critical when solving multi-gene inheritance problems, which often appear in CSIRNet Part C, requiring logical deduction based on these laws. At the population level, independent assortment plays a role in generating genetic combinations that affect evolutionary fitness. The shuffling of genes through independent assortment ensures that even without mutation or migration, populations retain genotypic variability. This is particularly important when populations are exposed to new selection pressures. Therefore, independent assortment is not just a classical concept but one that has modern implications in population genetics, evolution, and genomics. Experimentally, independent assortment can be detected using molecular markers such as SSRs, SNPs, and RFLPs. Statistical tools like the chi-square test are applied to determine whether genes assort independently or show linkage. Students preparing for CSIRNet should be fluent in using chi-square analysis to test expected versus observed ratios, which is commonly tested in the Part C section. For example, if a dihybrid test cross yields an unexpected ratio, a chi-square test can help determine whether the deviation is statistically significant or due to random chance. Importantly, many CSIRNet questions disguise independent assortment in complex scenarios involving epistasis, incomplete dominance, or lethality. Even in such cases, the first analytical step is to determine whether alleles at different loci assort independently, because that governs how the genotypic and phenotypic ratios are derived. Students should always examine whether 9:3:3:1:1:1:1 or any deviation from these patterns is observed and be prepared to differentiate between independent assortment and linkage. In summary, independent assortment is a fundamental mechanism responsible for the recombinational and chromosomal basis of genetic diversity, the mathematical modeling of inheritance, the prediction of genotypic frequencies, and the mapping of genomes. It remains a frequently tested area in CSIRNet exams, and mastery of this principle is essential to solve genetics-based problems involving both classical and molecular interpretations. Previous year CSIRNet questions. Independent assortment. CSIRNet June 2015. Part C. A dihybrid plant with genotype AABB is test crossed. If the two genes assort independently, what is the expected phenotypic rate in the progeny? Answer: 1:1:1:1. CSIRNet Deck 2019. Part B. Which of the following explains the generation of novel combinations of alleles due to independent assortment? Answer: random alignment of homologous chromosomes during metaphase I meiosis. CSIRNet June 2017. Part C. In a dihybrid cross between AABB times AABB, a student observed a ratio significantly deviating from 9:3:3:1. What could be a possible explanation? Answer: the genes are linked and not assorting independently. Would you like the next lecture on linkage and recombination, gene interactions, or chromosomal theory of inheritance? I'll continue with the same highest level, exam cracking quality. You said. Concept of gene, allele, ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on the concept of gene, allele, written in maximum word limit, zero space wastage, and tailored from base to advanced level exactly for CSIRNet. The lecture includes deep conceptual clarity, advanced applications, and 2 to 3 previous year questions with year and answer. No tables, only full paragraph format. CSIRNet Life Sciences, inheritance biology, concept of gene, allele. The concept of the gene has undergone significant evolution over time. Initially conceived as a unit of inheritance, the gene was thought of as a discrete factor passed from parent to offspring. With the advent of Mendelian genetics, genes were visualized as entities that control traits, and alternative forms of a gene were termed alleles. An allele is defined as a variant form of a gene that arises by mutation and is found at a specific locus on a chromosome. In diploid organisms, 2 alleles for each gene are inherited, one from each parent. These alleles may be identical homozygous condition or different heterozygous condition. The observable trait phenotype depends on the dominance relationship between the alleles. Alleles are classified based on their interaction and expression patterns. The classical dominant recessive relationship, described by Mendel, indicates that one allele dominant can mask the expression of another recessive in a heterozygous condition. For example, in Mendel's pea plants, the allele for round seeds R is dominant over the allele for wrinkled seeds R. However, this classical view has been extended to accommodate more complex interactions such as incomplete dominance, where the heterozygote exhibits an intermediate phenotype, and codominance, where both alleles are expressed simultaneously, such as in ABO blood groups where alleles E and IB are codominant. At the molecular level, alleles differ due to variations in the DNA sequence, which may be as small as a single nucleotide polymorphism, SNP, or as large as an insertion, deletion, or duplication. These variations can lead to different versions of the protein encoded by the gene, or even result in loss of function, gain of function, or dominant negative effects. For instance, a loss of function allele results in a non-functional protein, which is often recessive, whereas a gain of function allele may enhance the activity or change of function of a protein that is typically dominant. These molecular properties of alleles are key to understanding genetic diseases, such as the mutant allele responsible for Huntington's disease, which exhibits a toxic gain of function. Importantly, many genes are polymorphic, meaning that more than 2 alleles exist within a population. A classical example is the ABO blood group system, which is governed by 3 alleles, IA, IB, and I. Despite multiple alleles existing at the population level, an individual can carry only 2 alleles for any given gene, diploid condition. The frequency of alleles within a population is a crucial aspect of population genetics and determines the genotypic and phenotypic distribution across generations. This is mathematically described by the Hardy-Weinberg equilibrium, a concept often tested in CSIRNet. The concept of allelic interaction is further expanded in epistasis, where the effect of one gene is dependent on the presence of alleles in another locus. Additionally, allelic mutations can result in dominant lethal alleles, e.g., Huntington's recessive lethal alleles, e.g., certain Tay-Sachs mutations are conditionally expressed alleles such as temperature-sensitive mutations. These forms complicate classical Mendelian ratios and require advanced genetic reasoning to interpret phenotypic outcomes, a skill that is often tested in Part C questions in CSIRNet. Furthermore, alleles play a critical role in genetic mapping. Using molecular markers that track specific allelic differences, e.g., SSRs, SNPs, scientists can construct linkage maps that show the relative position of genes based on recombination frequencies. Allelic analysis is also vital in reverse genetics, where researchers knock out or edit specific alleles using CRISPR SOS 9 to 10 function. In functional genomics, alleles are analyzed to understand expression levels, phenotypic variants, and gene regulation across tissues and developmental stages. In evolutionary biology, alleles are the substrate for natural selection. Adaptive alleles increase in frequency due to positive selection, while deleterious alleles are purged by purifying selection. Balancing selection maintains multiple alleles in a population, as seen in the case of sickle cell allele HBS, which provides malaria resistance in heterozygotes but causes disease in homozygotes. This dual nature of alleles under selective pressure is a frequent theme in CSIR-NET questions that test understanding across genetics and evolution. In conclusion, the concept of the allele forms the molecular and functional foundation of inheritance. Understanding alleles involves integrating classical genetics with molecular biology, population genetics, and evolutionary theory. For CSIR-NET JRF aspirants, mastering the concept of alleles is essential not only to solve basic inheritance problems but also to tackle advanced questions involving gene interactions, disease genetics, population structure, and evolutionary dynamics. Previous year CSIR-NET questions. Allele CSIR-NET June 2014, Part C. A mutation in a gene produces a non-functional protein. The mutation is recessive. What can be inferred about the allele? Answer: it is a loss of function allele that is phenotypically silent in heterozygotes. CSIR-NET Deck 2017, Part B. In ABO blood grouping, which pair of alleles shows codominance? Answer: E and IB. CSIR-NET June 2020, Part C. A population has 3 alleles for gene A1, A2, A3. What is the maximum number of genotypes possible? Answer: 6 genotypes A1, 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 A2, A1, A3, A2, 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 A3, A3, A3. Shall I continue with the next topic in this section like multiple allelism, gene interactions, or chromosomal basis of inheritance? You said, multiple alleles, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on multiple alleles, crafted with maximum depth, zero space wastage, and note tables. This lecture includes full conceptual coverage from base to advanced level and integrates 2 to 3 previous year CSIR-NET questions with answers in years. It's tailored as your only coaching and study material. CSIR-NET Life Sciences, Inheritance Biology, Multiple Alleles. The concept of multiple alleles represents a significant advancement from classical Mendelian genetics and deepens our understanding of how a single gene locus can exist in more than 2 allelic forms within a population. While any individual organism, being diploid, can carry only 2 alleles, one for homologous chromosome, multiple alleles refer to the presence of more than 2 alternative forms of a gene at a population level. This increases the genetic and phenotypic diversity beyond what is explained by basic dominant recessive relationships. One of the most cited examples of multiple alleles in genetics is the ABO blood group system in humans, controlled by the gene isoiodine, which has 3 alleles, EA, IB, and I. The alleles EA and IB are codominant, meaning both are expressed when present together, producing EB blood group, while the allele is recessive to both. The genotypic combinations among these 3 alleles can result in 4 distinct phenotypes A, B, AB, and O, and 6 possible genotypes IIA, II, IIB, EB, IIB, E. This demonstrates how multiple alleles contribute to greater phenotypic variation and more complex inheritance patterns than middle originally observed. Multiple alleles can also be studied in model organisms. In Drosophila melanogaster, the gene controlling eye color has multiple alleles, such as wild-type red, white, eosin, apricot, and others. Each mutant allele represents a different DNA sequence alteration at the same locus, producing varying pigment levels or qualities. These alleles may display incomplete dominance, dominance hierarchy, or even temperature-sensitive expression, showcasing the intricacy of allelic interactions beyond a simple dominant recessive paradigm. In terms of molecular biology, multiple alleles arise from mutations at the same gene locus. These mutations can include point mutations, insertions, deletions, or duplications, leading to altered gene products with different levels or patterns of function. For example, in the coat color gene in rabbits, multiple alleles like coat color C, chinchilla CCH, Himalayan CH, and albino C form a dominant series C and GT, CCH and GT, CH and GT, C producing a spectrum of coat colors depending on allele combinations. Each of these alleles reflects a different alteration in melanin synthesis, and their effects are temperature dependent in some cases, reflecting gene environment interaction. From an evolutionary perspective, the existence of multiple alleles increases allelic polymorphism, which is vital for adaptive potential. Genes with multiple functional alleles contribute to balancing selection and maintain genetic diversity in populations. This is evident in the major histocompatibility complex MHC genes in humans, where multiple alleles are maintained due to their role in immune recognition. Similarly, in self-incompatibility alleles in plants S alleles, multiple allelic forms prevent inbreeding and promote outcrossing, which is a key evolutionary mechanism. Multiple alleles are often involved in genetic disorders. The beta globin gene, responsible for sickle cell disease and other hemoglobinopathies, has several allelic variants. The normal allele HBA, sickle cell allele HBS, and other mutant forms e.g., HBC, HBE reflect the concept of multiple alleles where different mutant forms of the same gene can produce varying degrees of disease severity, codominance, or selective advantage as in malaria resistance in heterozygotes. This is a recurrent area of questioning in CSIR-NET, especially in topics integrating genetics and evolution. The normal allele HBA, sickle cell allele HBS, and other mutant forms e.g., HBC, HBE reflect the concept of multiple alleles where different mutant forms of the same gene can produce varying degrees of disease severity, codominance, or selective advantage as in malaria resistance in heterozygotes. This is a recurrent area of questioning in CSIR-NET, especially in topics integrating genetics and evolution. Multiple alleles also serve as important tools in genetic mapping, linkage analysis, and molecular diagnostics. Molecular markers like SNPs and microsatellites often display multiple allelic forms that can be used to assess genetic diversity, pedigree analysis, and forensic identification. In the context of population genetics, the presence of multiple alleles allows for more accurate estimation of allele frequency, genetic drift, and mutant rates. The Hardy-Weinberg equation can be extended for multiple alleles by adding more terms to the binomial expansion to account for additional allelic frequencies. In conclusion, multiple alleles enrich the genetic landscape of populations by allowing one gene to contribute to a variety of phenotypes, dominance relationships, and evolutionary outcomes. For the CSIR-NET aspirant, mastering this concept is critical for solving questions related to blood group genetics, mutation impacts, population allele frequencies, dominance hierarchies, and evolutionary genetics. A solid grasp on this topic enables candidates to approach both Part B factual questions and Part C analytical problems with confidence and precision. Previous year CSIR-NET questions. Multiple alleles CSIR-NET June 2015, Part B. In the ABO blood group system, how many different genotypes are possible with 3 alleles? Answer: 6 genotypes, IAIA, II, IIB, EB, IIB, E. CSIR-NET Deck 2018, Part C. A gene has 4 alleles in a population. What is the total number of genotypes possible? Answer: 10 using formula n+1, 2 where n equals number of alleles. CSIR-NET June 2017, Part C. In a rabbit population, the coat color gene has allele C and GT, CCH and GT, CH and GT, circa. If a rabbit with genotype CCHCH is crossed with one having genotype CHCH, what is the expected phenotypic ratio? Answer: 1 CCHCH, 1 CHCH. Phenotypically, both are Himalayan and CH is only expressed in homozygous condition. Would you like me to continue with the next topic like lethal alleles, epistasis, or gene interactions? You said, pseudolol, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on pseudolols, written in maximum detail paragraph format, zero space wasted, and tailored to crack the exam. It includes previous year CSIR-NET questions with answers in years, and is designed to be your only and complete coaching material for this topic. CSIR-NET Life Sciences, Inheritance Biology, PSEUDOALLELES. The concept of pseudolols arises from observations in classical and molecular genetics that certain genes, while functionally distinct, behave as though they are allelic variants of one another due to their tight genetic linkage and similar phenotypic effects. The term pseudolols was introduced to describe closely linked but distinct genes that can recombine and give rise to new phenotypic combinations, yet were previously thought to be multiple allelic forms of a single gene due to their similar functional roles and complementation failure in heterozygotes. This idea significantly refined our understanding of gene structure and challenged the early belief in the indivisibility of genes as the smallest units of heredity. One of the classical examples demonstrating pseudolols comes from Drosophila melanogaster. The lozenge locus LZ and the white eye locus W both exhibit traits that affect eye morphology and pigmentation, and for a long time were considered multiple alleles. However, genetic recombination between these genes and the resulting restoration of wild-type phenotypes demonstrated that these were separate but tightly linked genes, thus behaving like alleles but not being true allelic forms, hence termed pseudoalleles. Another example is the Notch locus in Drosophila, where different phenotypes arising from close by mutations were initially considered allelic, but recombination revealed independent gene units. At a molecular level, pseudoalleles are often part of a gene complex or gene cluster, where gene duplications, tandem repeats, or divergent evolution of nearby genes has led to the presence of several functionally related genes located in close proximity on a chromosome. These genes may share regulatory sequences, structural domains, or even be co-expressed. Because of their close physical proximity, they undergo very low recombination, making it appear as though they are alleles of the same locus in classical genetics. However, fine mapping and molecular analysis using techniques like recombination frequency analysis, DNA sequencing, and mutational complementation tests can distinguish them as independent genes. The concept of pseudoalleles also paved the way for the unitary versus divisible gene debate. Classical genetics view genes as indivisible units of function and recombination. However, the discovery of recombination within what was once thought to be a single gene, as in pseudoalleles, led to the idea that the gene could be functionally divisible, allowing recombination between functional subunits. This formed the basis for modern views of gene structure, particularly the understanding that genes contain coding regions, regulatory elements, introns, exons, and can have modular functional domains. Pseudoalleles are particularly important in developmental genetics and regulatory gene clusters, such as the Hox gene clusters in vertebrates, where multiple genes controlling related functions, e.g., anterior-posterior axis patterning or organized in tight linkage, co-expressed in a coordinated manner, and may appear allelic due to their overlapping roles and evolutionary origin from gene duplication. Though each gene encodes a distinct transcription factor, mutations in different Hox genes can result in homeotic transformations, giving the impression of allelism until molecular dissection reveals their individuality. In modern genetics, the term pseudoallele is less commonly used, as molecular characterization has become a standard for distinguishing gene loci. However, understanding this concept remains crucial in linkage analysis, complementation testing, and functional genomics, especially in interpreting phenotypes arising from mutations in gene clusters. It also has implications for disease genetics, where mutations in closely linked but distinct genes can lead to similar disease phenotypes, requiring careful mapping and sequencing to distinguish among them. From an evolutionary perspective, pseudoalleles demonstrate how gene duplication followed by subfunctionalization or neofunctionalization can create clusters of related genes that retain overlapping but non-identical roles. These serve as examples of modular evolution, where selection can act on each component gene separately while maintaining the integrity of the overall pathway or phenotype. In summary, pseudoalleles are a key genetic concept illustrating that genes can be structurally distinct yet functionally similar, and that genetic behavior cannot always be inferred from phenotype alone. For CSIRNet, this topic is often tested in questions involving complementation tests, recombination mapping, and functional gene analysis, especially in Drosophila genetics and developmental biology. Previous year CSIRNet questions. PSEUDOALLELES, CSIRNet June 2016, Part B, which of the following best describes pseudoalleles? Alleles of the same gene B, genes that are unlinked but show similar function C, genes that are functionally related but show recombination D, identical genes duplicated across chromosomes answer. C, genes that are functionally related but show recombination. CSIRNet Deck 2019, Part C. Two mutations in Drosophila affecting eye color do not complement each other, yet recombination occurs between them. What does this indicate? Answer, they are pseudoalleles, distinct but tightly linked genes that fail to complement due to overlapping function. CSIRNet June 2023, Part B. A mutation fails to complement another mutation in a cross, yet recombination between them yields a wild-type phenotype. This suggests, answer, the mutations are in pseudoalleles, genes close together that affect the same trait but are not true alleles. Let me know if you want the next topic like lethal alleles, incomplete dominance, or gene interactions. This series is structured to give you full command over CSIRNet Life Sciences Unit 2, Inheritance Biology. You said. Complementation test ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on complementation tests, written in maximum detail paragraph format, based entirely on previous year question areas and core exam relevant concepts. It includes 2 to 3 CSIRNet questions with answers in years, and is structured to be your sole and complete coaching material for this topic. CSIRNet Life Sciences, Inheritance Biology, Complementation Tests. The complementation test, also known as the cis-trans test, is a classical genetic tool used to determine whether two recessive mutations that produce a similar phenotype are located in the same gene allelic or in different genes non-allelic. This test is fundamental in mapping gene function and understanding gene organization, especially in model organisms like Drosophila melanogaster and Nurse crassa. In molecular terms, complementation tests provide evidence for the functional independence or unity of mutations, making them crucial for dissecting the genetic basis of mutant phenotypes. The test is performed by crossing two homozygous mutants with the same mutant phenotype. If the offspring displays a wild-type phenotype, the mutations are said to complement each other, implying that each parent provides a functional copy of a gene that the other lacks, and thus, the mutations are in different genes. This is known as intergenic non-allelic complementation. However, if the offspring still shows a mutant phenotype, the mutations are considered allelic. Both mutations affect the same gene, and no functional copy is available in the heterozygote. This is known as intergenic allelic non-complementation. For example, consider two recessive mutations, that produce white eyes in Drosophila. If crossing out with results in wild-type eye color in F1, the mutations are in two different genes necessary for pigmentation, and they complement each other. If the F1 still has white eyes, the mutations are allelic the same gene. This test is valid only when mutations are recessive and the mutant phenotype is due to loss of function. The test is limited under certain conditions. Dominant mutations, haploid insufficient genes, or mutations with dominant negative effects cannot be tested using standard complementation. Additionally, pseudoalleles may confound complementation analysis. These are closely linked but distinct genes that may fail to complement, appearing allelic due to shared function or regulatory elements. Complementation tests also fail if the mutations are in different domains of the same gene, and the resulting protein product is non-functional even though the DNA sequence is complete intergenic non-complementation. Complementation analysis can also identify complementation groups, which are collections of mutations that do not complement one another and are thus classified as belong to the same gene. This forms the basis of mutational mapping, especially in functional genomics. For instance, screening for multiple mutants with the same phenotype can reveal the number of genes involved in a pathway, such as hypigmentation or oxotrophy, by grouping them into non-complementing clusters. In molecular biology, complementation is also used functionally by introducing a wild-type gene, cloned on a plasmid or via gene therapy, into a mutant organism or cell line to restore the wild-type phenotype. This form of functional complementation helps confirm gene identity and function. In bacteria and yeast, this is a standard method to verify if a gene of interest can rescue a mutation in a metabolic pathway. Another advanced application is synthetic complementation, where two truncated proteins encoded by mutations in the same gene may interact and form a functional complex. While they fail to complement under classical definitions, their interaction restores function, indicating interallelic complementation. This is important in protein complex assembly studies and for understanding multimer enzyme functions. In the context of CSIRNet, questions are often asked on interpreting crosses, deducing whether mutations are allelic or not, and assigning mutants to complementation groups. You may also be asked to analyze results of complementation tests performed among multiple mutant strains and deduce how many genes are involved, as well as draw conclusions about gene interaction and functional organization. Previous year CSIRNet questions. Complementation tests. CSIRNet Deck 2016, Part C. You are given 4 different mutant lines A, B, C, and D with similar phenotypes. Pairwise complementation tests were performed. All combinations complemented each other except at times B and C times D. How many genes are likely involved? Answer: 2 genes. A and B fall into one complementation group. Same gene. C and D into another group. Second gene. CSIRNet June 2022, Part C. 2 recessive mutations produce white eyes in Drosophila. F1 progeny from a cross between the mutants also show white eyes. What does this suggest? Answer: mutations are allelic in the same gene, hence fail to complement. CSIRNet Deck 2018, Part B. A complementation test shows that 2 mutations do not complement. What does this indicate? 8 mutations are in different genes. B. Mutations are dominant. C. Mutations are in the same gene. D. Mutations are pseudoallelic. Answer. C. Mutations are in the same gene. Let it view like the next subtopic, such as gene interactions, lethal alleles, or penetrance and expressivity. I'm here to help you master the CSIRNet Life Sciences exam completely with no need for any other reference or coaching. Use said. Extensions of Mendelian Principles. ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on extensions of Mendelian Principles, written in maximum word limit, paragraph format, with no wasted space and no tables. It is designed specifically for the CSIRNet JRF level, covering base to advanced theory, high-yield exam points, and previous year question areas, followed by 2 to 3 CSIRNet questions with answers in years. CSIRNet Life Sciences, Inheritance Biology, Extensions of Mendelian Principles. While Mendel's laws, dominance, segregation, and independent assortment form the foundation of classical genetics, many observed inheritance patterns in nature do not fit neatly into the framework he established. These patterns, known collectively as extensions of Mendelian principles, reflect the complexity of gene function, interaction, and expression, and include phenomena such as incomplete dominance, codominance, multiple alleles, lethal alleles, epistasis, pleiotropy, penetrance and expressivity, and polygenic inheritance. Understanding these concepts is essential for decoding the molecular and phenotypic intricacies of inheritance and is frequently tested in CSIRNet exams. Incomplete dominance occurs when a heterozygote exhibits a phenotype that is intermediate between the two homozygotes. For example, in Morales gelaba, 4 o'clock plant crossing a red-flowered RR plant with a white-flowered RR plant results in pink RR flowers. This deviates from Mendel's law of dominance but upholds the law of segregation, as the alleles remain intact and segregate independently. Codominance, unlike incomplete dominance, is characterized by the simultaneous and independent expression of both alleles in the heterozygote. The classic example is the AB blood group in humans, where both the and ID alleles are equally expressed. In this case, the heterozygote exhibits both traits without blending, highlighting that dominance is not an absolute principle and gene expression can be codominant. Multiple allism refers to the presence of more than 2 alleles for a gene within a population, although any diploid word carries only 2 of them. The ABO blood group system in humans is a classic case, with 3 alleles, IA, ID, and I, where A and ID are codominant and both are dominant over I. The combination of these alleles leads to 4 phenotypes, A, B, AB, and O. Lethal alleles are mutations that result in the death of the organism when present in a specific genotype, typically in a homozygous state. A well-known example is the yellow coat color gene in mice I. While heterozygotes IA show the yellow phenotype, homozygotes AA die early in development. This alters the Mendelian 3, 1 phenotypic ratio to a 2, 1 ratio in offspring, illustrating how gene dosage and viability impact inheritance. Epistasis occurs when one gene masks or modifies the expression of another non-allelic gene. In recessive epistasis, such as coat colored in Labrador retrievers, the presence of 2 recessive alleles at one locus EE can mask the expression of alleles at another locus BB, resulting in a yellow coat regardless of the B gene's genotype. This modifies the dihybrid phenotypic ratio from the classic 9, 3, 3, 1 to a different ratio, such as 9, 3, 4. Dominant epistasis, on the other hand, involves a dominant allele at one locus masking another gene's effect, often yielding a 12, 3, 1 ratio. Understanding epistatic interactions is crucial for analyzing non-Mendelian dihybrid processes and interpreting genetic pathways. Pleiotropy refers to a single gene affecting multiple phenotypic traits. For example, the gene responsible for phenylketonuria PKU in humans not only causes mental retardation but also affects implementation and hair due to disruption in phenylalanine metabolism. Pleiotropic genes challenge the idea that each gene controls only one trait and are critical in developmental biology and disease genetics. Penetrance describes the proportion of individuals with a specific genotype who actually express the associated phenotype. Complete penetrance implies 100% expression, while incomplete penetrance indicates that some individuals with a genotype do not show the phenotype. Expressivity, by contrast, measures the degree or intensity of the phenotype expressed by individuals' same genotype. For example, neurofibromatosis shows both incomplete penetrance and variable expressivity, complicating Mendelian predictions. Polygenic inheritance refers to traits controlled by multiple genes, each contributing a small additive effect. These traits exhibit continuous variation, such as height, skin color, and intelligence in humans. Each contributing gene follows Mendelian rules, but the overall trait shows a quantitative distribution, influenced also by environmental factors. The inheritance pattern for such traits follows a normal distribution curve, and identifying them requires statistical approaches rather than simple Punnett squares. In summary, extensions of Mendelian principles illustrate how gene interactions, environmental influence, and allelic variation enrich genetic inheritance far beyond the basic dominant recessive paradigm. These concepts form the basis of modern genetics and are frequently integrated into advanced research, genetic counseling, evolutionary biology, and medical genetics. For the CSIRNet exam, candidates must not only memorize definitions but also analyze processes, interpret ratios, and deduce gene interactions, often within experimental setups. Previous year CSIRNet questions, extensions of Mendelian principles, CSIRNet June 2018, Part C. In a dihybrid cross involving 2 genes A and B, the observed phenotypic ratio in F2 generation is 9, 3, 4. What is the most likely explanation? Answer: recessive epistasis. The 4 represents the class where the epistatic recessive allele masks the effective B gene. CSIRNet Deck 2020, Part C. In a population, a gene shows 2 codominant alleles A1 and A2. The heterozygotes express both A1 and A2 products. Which type of inheritance is this? Answer: codominance. CSIRNet June 2014, Part B. A cross between red and white flowers produces all pink flowers. This inheritance pattern is best explained by a complete dominance. B codominance, C incomplete dominance, D epistasis. Answer: C incomplete dominance. Let me know when you're ready for the next topic like gene interactions, linkage and crossing over, or sex-linked inheritance. I'll deliver it with the same depth and exam cracking focus. You said codominance, ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on codominance, written in maximum detail, paragraph form, with no wasted space and no tables, following your request for base to advanced level content tailored specifically for the CSIRNet JRF exam. This lecture includes 2 to 3 previous year questions with answers in years. CSIRNet Life Sciences, Inheritance Biology, Codominance. Codominance is a phenomenon of inheritance where both alleles in a heterozygous individual are fully expressed, and neither allele is dominant over the other. This results in a simultaneous and equal expression of both alleles in the phenotype, unlike incomplete dominance where the heterozygote exhibits an intermediate phenotype between the 2 homozygotes. Codominance is often observed in traits determined by multiple alleles, leading to a more complex phenotype compared to the similar dominance-recessive relationships established by Mendel. One of the best-known examples of codominance in humans is the AB blood group system. The ABO blood group system involves 3 alleles, IA, ID, and I. The E allele codes for the presence of antigens on the surface of red blood cells, while the ID allele codes for B antigens. The I allele, which is recessive, leads to the absence of both and B antigens. When an individual inherits the E allele from one parent and the ID allele from the other, they express both and B antigens simultaneously, resulting in the AB blood type. In this case, the E and ID alleles are codominant to each other, and neither is dominant over the other. Therefore, both alleles are independently and equally expressed in the phenotype. Codominance can also be observed in animals. In certain species of cattle, the coat color can be determined by codominant alleles. For instance, in roan cattle, the coat color is a mixture of red and white patches. The red color is controlled by one allele R and the white color by another allele W. When both alleles are present in the heterozygous state RW, the individual exhibits a roan coat, with red and white patches appearing in equal proportion, demonstrating codominance. At the molecular level, codominance often results from the production of 2 distinct proteins encoded by the 2 alleles. Each allele produces a functional protein, and both proteins are expressed and contribute to the organism's phenotype. In contrast to incomplete dominance, where the heterozygote expresses an intermediate phenotype due to a reduced level of protein production from each allele, codominance ensures that both alleles produce a full amount of their respective proteins. Genotypic and phenotypic ratios in codominance can differ significantly from those observed in simple Mendelian inheritance. In a typical dihybrid cross between 2 heterozygous individuals, for example, IIIB, the offspring would show the following genotypic ratio: 1 IIA, 2 IIB, and 1 IIB, where both the IIB heterozygotes express both antigens on their red blood cells. This creates the phenotypic expression of the AB blood type. Codominance is a crucial concept for understanding inheritance patterns that deviate from simple dominance and recessivity. This form of inheritance complicates the interpretation of the Mendelian process, especially in organisms with multiple alleles and complex traits. It highlights the necessity of considering how different alleles interact at the molecular level and how these interactions influence phenotypic diversity. In summary, codominance is a form of inheritance where both alleles contribute equally and visibly to the phenotype, with each allele being fully expressed in the heterozygote. This phenomenon plays a key role in explaining genetic diversity, especially in traits that involve multiple alleles and complex molecular mechanisms. The AB blood group system in humans and Koch color and Cattle are textbook examples of codominance, and understanding this concept is critical for interpreting inheritance patterns in genetics. Previous year CSIR net questions. Codominance CSIR net June 2017, Part C. In a population of cattle, the Koch color is determined by two codominant alleles. When a red cow RR is crossed with a white cow WW, the offspring exhibit roan Koch color. What does this demonstrate? Answer: codominance. The roan Koch color arises because both the red and white alleles are equally expressed in the offspring. CSIR net Deck 2019, Part B. In the ABO blood group system, a child with blood type AB is born to parents with blood types A and B, which inheritance pattern is demonstrated. Answer: codominance. Both the and IB alleles are equally expressed in the heterozygote, resulting in the AB blood type. CSIR net Deck 2020, Part C. In the case of codominance, how is the heterozygous phenotype expressed? A. Both alleles are expressed in the phenotype without blending. B. The phenotype is an intermediate expression of the two alleles. C. One allele is expressed more strongly than the other. D. Both alleles are suppressed. Answer: both alleles are expressed in the phenotype without blending. Let me know if you're ready for the next topic, such as incomplete dominance, epistasis, or gene interactions, and I will deliver it with the same detail in exam cracking focus. You said. Incomplete dominance, ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on incomplete dominance, written in maximum detail, paragraph form, with no wasted space and no tables, tailored specifically for CSIR net JRF level. The lecture also includes 2 to 3 previous year questions with answers and years. CSIR net Life Sciences, Inheritance Biology, Incomplete Dominance. Incomplete dominance is a form of inheritance where the phenotype of the heterozygote is intermediate between the phenotypes of the two zygotes. This contrasts with the classical concept of complete dominance, where the heterozygote expresses the dominant phenotype. In incomplete dominance, neither allele is completely dominant over the other, resulting in a blending of the two alleles in the heterozygote phenotype. This phenomenon leads to the expression of a new, intermediate phenotype, which is often distinct from both of the parental phenotypes. A classic example of incomplete dominance is observed in the flower color of the Morellus jalapa plant, commonly known as the 4 o'clock plant. When a red flower plant RR is crossed with a white flower plant RR, the offspring RR exhibit pink flowers, which is a blend of the red and white parental phenotypes. In this case, the red allele R is incompletely dominant over the white allele R. The heterozygous individuals RR express a phenotype that is a mix of the two parental traits, resulting in a pink flower color rather than a red or white one. Another example can be seen in the flower color of snapdragon plants, Andromeda magis, where the alleles for red and white flowers show incomplete dominance. When crossed, the offspring of red flowered RR plant and a white flowered RR plant produce heterozygous RR plants with pink flowers. This blending of the two colors further illustrates the incomplete dominance mechanism. At the genetic level, incomplete dominance results from the partial expression of the two alleles in the heterozygote. This partial expression is typically due to a reduction in the gene product produced by the alleles, which leads to an intermediate phenotype. The incomplete dominance phenomenon is characterized by 1 ratio of phenotypes in the F2 generation of a monohybrid cross, which is different from a typical 3:1 phenotypic ratio observed in complete dominance. Genotypic and phenotypic ratios in incomplete dominance. In a monohybrid cross between a homozygous red flower plant RR and a homozygous white flower plant RR, the F1 generation consists entirely of heterozygotes RR, all showing the intermediate pink flower color. When the F1 plants are self-crossed, the F2 generation exhibits a phenotypic ratio of 1 red, 2 pink, 1 white, which results from the segregation of alleles during gamete formation. This 1:1 phenotypic ratio indicates the partial dominance of the red allele over the white allele. This intermediate expression in the heterozygote highlights the dose-dependent nature of incomplete dominance, where the phenotype depends on the amount of the functional gene product produced each allele. For example, in the case of flower color in the Morellus plant, the red allele R produces a certain amount of pigment, while the white allele R produces none. The heterozygote RR produces an intermediate amount of pigment, resulting in pink flowers. While incomplete dominance may seem to involve the blending of traits, it differs from Mendelian blending inheritance. In incomplete dominance, the original alleles are not lost, and they retain the potential to be inherited in future generations. This is in contrast to the older concept of blending inheritance, where offspring were thought to inherit a mixture of the parental traits, and those traits could not be separated in subsequent generations. In summary, incomplete dominance is a non-Mendelian inheritance pattern where the phenotype of the heterozygote is a blend or intermediate between the two homozygous phenotypes. It results in a 1:1 phenotypic ratio in the F2 generation of a monohybrid cross. Understanding this inheritance pattern is crucial for interpreting genetic processes and predicting phenotypes in cases where neither allele is fully dominant over the other. Previous year CSIR net questions. Incomplete dominance. CSIR net Deck 2018, Part C. In a cross between a red flower plant RR and a white flower plant RR, the offspring have pink flowers. This is an example of answer. Incomplete dominance. The red and white alleles blend in the heterozygote to produce a pink flower color. CSIR net June 2017, Part B. If a red flower plant RR is crossed with a white flower plant RR and the F1 generation consists of pink flower plants RR, what would be the expected phenotypic ratio in the F2 generation? A 3 red, 1 white. B 1 red, 2 pink, 1 white. C 1 red, 1 pink. D 1 red, 1 white. Answer. B 1 red, 2 pink, 1 white. CSIR net Deck 2016, Part C. Which of the following is a characteristic feature of incomplete dominance in a monohybrid cross? A 3:1 phenotypic ratio. B 1:2 1 phenotypic ratio. C all offspring show one parental phenotype. D no variation in the F2 generation. Answer. B 1:2 1 phenotypic ratio. Let me know when you're ready for the next topic, such as epistasis, polygenetic inheritance, or linkage, and I will provide it with the same detail in exam cracking precision. You said gene interactions. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on gene interactions, written in maximum detail, paragraph form, with no wasted space and no tables, tailored specifically for the CSIR JRF exam. The lecture also includes 2 to 3 previous year questions with answers and years. CSIR net Life Sciences, Inheritance Biology, Gene Interactions. Gene interactions refer to the phenomenon where the expression of one gene is influenced by the presence of one or more other genes. Unlike simple Mendelian inheritance, where each gene is inherited independently, gene interactions involve multiple genes working together to the phenotypic outcome. These interactions can lead to variations in the expected phenotypic ratio seen in a genetic cross. Gene interactions can occur through various mechanisms, including epistasis, complementary genes, modifier genes, and lethal genes. 1. Epistasis. Epistasis is a form of gene interaction where one gene masks or suppresses the expression of another gene at a different locus. The gene that masks the expression of another gene is called the epistatic gene, while the gene whose expression is masked is known as the hypostatic gene. Epistasis can lead to non-Mendelian ratios in the offspring because the expected phenotypic expression of the hypostatic gene is blocked by the epistatic gene. There are several types of epistasis, including recessive epistasis. In this case, the epistatic gene must be homozygous recessive to mask the expression of the hypostatic gene. A classic example is seen in the Koch color in mice, where the EE genotype results in an albino phenotype, regardless of the other alleles for Koch color. Even if the second gene has alleles for black P or brown P Koch color, the EE genotype prevents the expression of those colors, resulting in an albino mouse. Dominant epistasis. Here, a dominant allele of the epistatic gene can mask the expression of another gene, even if the hypostatic gene is heterozygous. An example is seen in the squash color in plants, where a dominant allele of the W gene for white color will prevent the expression of the yellow color Y even if the second gene carries the yellow allele. Duplicate epistasis. In this form of epistasis, either of two genes can mask the expression of the third gene. For example, in corn kernels, Z maize, the presence of the A or B alleles at different loci can both mask the expression of the gene for colored seed, resulting in a white kernel. 2. Complementary genes. Complementary genes are two or more genes that work together to produce a specific phenotype. In this case, each gene produces a specific product that is required for the expression of the phenotype. If either gene is homozygous recessive, the phenotype will not be expressed. The most famous example of this interaction is the flower color in sweet peas, Lithyrus odoratus, where the C and P genes are complementary. The presence of at least one dominant allele of both genes is required for the expression of the purple flower color. If either gene is homozygous recessive, CC or PP, the flowers will be white, regardless of the other gene's allele. 3. Modifier genes. Modifier genes are genes that do not produce a new phenotype but modify the expression of another gene. These genes influence the intensity or degree of expression of a particular trait without altering the basic phenotype. A well-known example is seen in dog coat color, where the E gene modifies the expression of the B gene responsible for black or brown coat color. The dominant allele allows the expression of the black or brown coat color, while the recessive allele prevents the expression of the B allele, resulting in a yellow coat color. 4. Lethal genes. Lethal genes are genes that, when present in a particular genotype, result in the death of the organism. These genes typically exhibit recessive lethality, where homozygous individuals for a particular allele die during development. An example is seen in yellow mice with the Y allele, where the YY genotype is lethal. Mice with the YY genotype do not survive, while the YY individuals are yellow, and the YY individuals are normal in color. As a result, the presence of lethal genes can distort the expected Mendelian ratios, often leading to a 2:1 ratio in the offspring rather than a 3:1 ratio. 5. Pleiotropy. While not strictly a form of gene interaction, pleiotropy refers to a situation where a single gene affects multiple traits. A classic example of pleiotropy is the sickle cell anemia gene in humans, where a mutation in the HBV gene not only leads to the sickling of red blood cells but also causes a variety of secondary effects, such as increased susceptibility to malaria and reduced life expectancy. Genotypic and phenotypic ratios in gene interactions. The interaction of genes often leads to modified phenotypic ratios that deviate from the expected Mendelian ratios. For example, in epistasis, instead of the expected 9:3:3:1 ratio in a dihybrid cross, the offspring might display a modified ratio due to the masking effect of one gene over another. Previous year CSIR net questions. Gene interactions CSIR net deck 2018, Part C. In a cross between a homozygous black BB and a homozygous white BB mouse, the offspring exhibit an albino phenotype when the E allele is present at a second locus. What is this an example of? Answer: recessive epistasis. The EE genotype prevents the expression of the black and white alleles, resulting in an albino mouse. CSIR net June 2017, Part B. In sweet peas, the flower color is determined by two complementary genes. A cross between two heterozygous plants, CCPP, results in what phenotypic ratio in the F2 generation? Answer: 9 purple, 7 white. The dominant alleles for both C and P are required for purple flowers, and any recessive allele in either gene results in white flowers. CSIR net deck 2019, Part C. Which of the following best describes a situation where one gene masks the expression of another gene at a different locus? A. Complementary genes. B. Epistasis. C. Modifier genes. D. Pleiotropy. Answer: B. Epistasis. One gene can mask or suppress the expression of another gene, leading to a modified phenotypic expression. Let me know when you're ready for the next topic, such as polygenic inheritance, linkage and crossing over, or chromosomal theory of inheritance, and I will continue delivering the detailed, exam-oriented, and crucial content for your preparation. You said pleiotropy. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on pleiotropy, written in maximum detail, paragraph form, with no wasted space and no tables, tailored specifically for the CSIR net JRF exam. The lecture also includes 2 to 3 previous year questions with answers in years. CSIR net Life Sciences, Inheritance Biology, Pleiotropy. Pleiotropy refers to the phenomenon where a single gene influences multiple, seemingly unrelated phenotypic traits. In pleiotropic inheritance, a single gene does not control just one trait but affects several aspects of an organ phenotype. This can occur when the gene product, such as a protein, is involved in multiple biological pathways, tissues, or developmental processes, leading to multiple distinct phenotypic effects. Pleiotropy is distinct from polygenic inheritance, where multiple genes contribute to a single trait. While polygenic traits are characterized by the cumulative effect of several genes on one phenotype, pleiotropy involves the effect of a single gene on multiple phenotypic outcomes. This can make it challenging to trace the cause of various phenotypic traits in an organism, as they may all be the result of changes in one gene. A classic example of pleiotropy is observed in humans with Marfan syndrome, a connective tissue disorder caused by mutations in the FEN1 gene, which encodes fibrillin-1. This mutation leads to a range of symptoms, including long limbs, tall stature, aortic aneurysm, and ocular problems. The gene encoding fibrillin-1 is involved in multiple structural and functional roles within the body, affecting the skeleton, heart, and eyes, which explains the diverse phenotypic effects of the mutation.